All right, here we are again for another AP Biology review. This is the second part of the energy review. Last time was about enzymes and just energy in general. This one's going to be about photosynthesis and cellular respiration. This is one of the longer videos, I think, because this unit has a lot of information. I'm also going to utilize kind of like a concept map strategy on this one, which you can like follow along and draw the concept map with me or make your own or once you get the idea. They are kind of interesting ways to study by connecting ideas. In this case, we're going to be connecting like vocabulary words because there's a lot of vocabulary words. So we're just going to jump in there with photosynthesis. Photosynthesis, of course, the job of this chemically is to reduce carbon. Sometimes people would say the job is to fix carbon. Of course, the less chemistry way of saying that is that we're going to make some glucose, right? And so this is the process of making food. This diagram shows the whole overview. We looked at this quite a bit before. If you can explain what's happening in this picture, then you are well on your way to having all the information. There's, of course, more specific details, but this is the main ideas if you can explain this whole thing. So, of course, the whole part starts with light. That light energy is needed to perform something called the light reactions. So the light reactions happen on a thing called the thylakoid, right, which we talked about in the membrane video. This is where the chlorophyll is found, and the chlorophyll absorbs the light so that we can pass it through a bunch of reactions, the light reactions, and create ATP. We also want to harvest electrons in the form of NADPH. Those electrons have to come from somewhere, which is the water, right? So the water is going to give up its electrons, and in the process, we're going to make oxygen. The energy molecules are then going to be used to create sugar, right? The building blocks of the sugar are carbon dioxide. We need ATP and electrons to put all those carbon dioxides together to make sugar. This is all happening outside of the thylakoids in a place called the stroma. And then, of course, in this video, it's emphasizing the idea that all these empty energy molecules are going to go back in as part of this cycle to get re-energized by the light. Right? So that's the basic idea. One thing to start off with, though, besides just photosynthesis going into respiration, is that this is all about ATP. We must get ATP to run our bodies. So we make ATP by taking ADP, adenosine diphosphate, and adding another phosphate onto it to make adenosine triphosphate. There's two ways to do this. The simplest way is actually substrate level phosphorylation. This is one of those vocabulary words. I wanted you to like see it right there, which is where we take a substrate that has some sort of phosphate on it that is readily accessible. We're going to take the phosphate off and give it to the ADP to make ATP, as shown in the bottom diagram, and also in this top diagram, although this one shows it more specifically with the ADP with the 2 phosphates becoming ATP with the 3 phosphates, right? This is the most direct and easy way to make ATP, but it has a big flaw, which is that you need this. You need some sort of molecule that has a phosphate that's easily transferable, and that doesn't usually happen. In fact, in this particular diagram, both of these diagrams, I guess specifically this one, this molecule is something that you had to spend energy to actually make that molecule. So it's not the best way to make ATP. The best way to make ATP is with something called chemiosmosis, right? This is the generic term. Whether it's talking about photosynthesis or respiration, it is talking about chemiosmosis works for both of these. It describes the part where you take an electron transport chain to pump hydrogens into a space. Notice that there's some differences on the left and right hand side of this diagram. And once the hydrogens are in that space, the concentration gradient is established. We have a lot of H pluses out here, a very low amount inside, of course, because that's where we're taking them from. And the only way to get the concentration gradient to balance itself out is through this enzyme called ATP synthase, which spins around as the hydrogens flow, and that energy is used to take ADP and convert it into ATP, right? And so this is a very efficient, although complicated way of doing things. It relies on having lots and lots of hydrogens, which again, that's going to rely on getting lots and lots of electrons. So that's kind of what I'm going to show here, right? That ATP synthase is the key for chemiosmosis. We need hydrogen ions and all that good thing, and that comes from the electron transport chain, right? And so the electron transport chain, again, is something that's going to happen in both, ooh, that's flashing a lot, photosynthesis and respiration. We're going to talk about the photosynthesis one, as you can see. Now, this is a different representation. I like to show a lot of these different kinds of representations of photosynthesis because reading diagrams is important. So let's kind of orient ourselves here that we have light, right? And light is hitting these green things, which of course we know are chlorophylls. And the energy is going to make an electron come out. Oh, there they are, right? So the light hits here, and this is chlorophyll, and that energizes an electron. And that electron is going to flow through an electron the electron transport chain. That's going to push the hydrogens where they need to go. Here is the more specific picture for photosynthesis, right? And so here we have the light coming into this photosystem, and here's the electron transport chain right in the middle, the first of them, right? The hydrogens are getting pushed inside. They keep on getting more and more concentrated in there, and then they have to flow out some way through the ATP synthase, right? Chlorophyll is the key to starting the electron transport chain. We saw pictures of this before, but here's a little more detailed part that you have your thylakoids, and then you zoom in, and the purple part here is a protein that has all these chlorophyll molecules embedded with our magnesium in the center. And they're arranged in a group to do two things. One, to maximize the amount of chlorophylls that you can fit into one space, and because there's a lot of different ones, you shouldn't have to know any of these. If they ever were going to talk about these pigments, they would tell you about them. There's specific names. You should just know in general that there are lots of different pigments. Chlorophyll A and B are the main ones, and these carotenoids are kind of the helpers. They help because they can absorb different wavelengths of light. Notice these ones can get this kind of wavelength at about, what, 480, whereas the peak here is about 460 or 470, and down here it's about 430, right? And so these are the two main ones. You get a lot of nice energetic blue light, but you don't always have blue light, and so sometimes you want to get some of the lighter blue light. Of course, we do have the red and the orangish light that we get at sunset can be absorbed down on the other side of the spectrum. Those are still being done by the chlorophylls. Anyway, you don't have to know all the names. You just need to know that there's a bunch of them. They're arranged in this thing called a photosystem, where they can funnel energy from one to the next. So these are all actually going to be getting hit, right? And so every time they're all getting hit, they just keep passing the energy to the center where the main chlorophyll is. The main chlorophyll gives up an electron. We would call this an electron acceptor. You don't have to know the names. Again, most of the time, if this is going to come up, these are going to be diagrams that you're going to be looking at that show you these things. Once the electron has gone to the electron acceptor, it's going to go to the electron transport chain. But it depends on which photosystem you're at. This is the tricky part. Photosystem 2 comes first. The light comes in and hits photosystem 2. It makes the electron go into the electron acceptor, which then starts the chain. Now this electron transport chain is making ATP. This is very often something that gets brought up on the AP exam. Photosystem 2 is for ATP. That's the one that's going to push the hydrogens. Now that same electron is going to get re-energized from photosystem 1, and it's going to go through a shorter electron transport chain, which is going to lead towards the creation of NADPH, right? So the electron that started with water, which you can't see, it's supposed to be right there. The electron got passed and passed and passed and eventually ended up on NADPH, right? So now we have two molecules, ATP and NADPH, that can go off to wherever it needs to go. The water is going to get split to give the electrons to the chlorophyll. Again, that's where the oxygen comes from. Then we take NADP to make NADPH. Those letters, you shouldn't have to know them specifically. If you see those kinds, right, the NADH, NADPH, FAD will come across. Those are just electron carriers. So here again is the big overview. All this stuff is now going to go off to the Calvin cycle. And so again, I'm just kind of in my concept map looking at just the parts that have to do with ATP specifically. ATP synthase, electron transport chain. All of these things are in the service of making energy molecules. Now in the Calvin cycle, we know we're going to take carbon dioxide and make glucose. But we can't just do that straight away. We need a lot of reactions, each one that has its own enzyme. And it does one small step at a time. Again, this is only part of it, but you can see that we're taking a 6-carbon sugar and then we split it into 2 3-carbon sugars, and then we add, we have a phosphate there, and then we are adding more phosphates there. And then this is gonna set up the molecule for the next step, right? And all these enzymes that have their chain of events. Out comes sugar, not technically glucose. It's called G3P, right? We can use that to make all kinds of different sugars. Glucose, of course, the main one, but we can use G3P to make lipids and anything else that we want to make that happens to have 3 carbons that you need, right? Oh, and there's the slide that was going to show that kind of thing, right? So we can use this as the backbone for all kinds of other molecules. So that line down there at the bottom gets taken away, and in its place we have the Calvin cycle. The Calvin cycle gives us G3P, which gives us glucose, and there we have photosynthesis, right? We started with chlorophylls absorbing light, and we ended up with glucose. Of course, we have the carbon dioxide. So then cellular respiration is going to be the opposite, and a lot of the same things we just talked about are all going to come again, but they're going to come in the reverse order. Just like last time, chemically, this is called, going over the name of chemically, this is called oxidizing our food. We now, to get the energy out of these molecules, we're going to undo it. And if last time we gave them electrons, oxidizing means take the electrons away. We're going to take our food, strip off all the electrons, and use those electrons to make ATP, right? So here we have, again, this whole thing. And that went gray for a minute, and I forget why. The idea, I guess we're not ready for that yet, but substrate level phosphorylation is something that is going to happen a little bit here, but we still really just want to talk about the chemiosmosis. This is the main part of it. We still have our ATP synthase and electron transport chain, but things are different now. Chlorophyll isn't the beginning anymore, right? We now need electrons that we've stripped from our food, and that's going to be from NADPH, right? That went away for. There's also this thing called FADH2. Again, you see those similar types of letter combinations, and you'll know that those are electron carriers. Last time we started with water. This time we're going to end with water. This is where the oxygen that you breathe comes in. So here we have the diagram that was just like the one from photosynthesis. It looks very, very, very similar, but everything's working in reverse, right? Here we have NADH and FADH2, they're dropping off the electrons that go through the chain. Those combine with water to make, oxygen to make water, right? The whole purpose of that is to get a whole bunch of hydrogens that are going to flow out through our ATP synthase. In this diagram, I like it because it does show you that, like, here's the part that's called chemiosmosis. Here's the electron transport chain. And there is one name for this big thing called oxidative phosphorylation, using oxygen to help us phosphorylate the ATP, right? Now, again, those names are kind of interesting because the oxygen doesn't actually touch the ATP, but we need the oxygen to make this all happen. Now, going down here at the bottom, where do we get those electrons from? In the last time, we were using the electrons and we got them from the water, but now we're getting them from our food. So instead of the Calvin cycle, we're going to do something called the Krebs cycle, right? Like this is the reverse of what we had before. we are not going to make ATP, or we're not going to get the, we're not going to make the ATP. We're going to make NADH and FADH2. We're going to release CO2 instead of use it. The G3P is called pyruvate now, and instead of glucose being the end, now glucose is the beginning, right? Our food is going to be broken down in the Krebs cycle to make electrons that we send through the chain. We eventually breathe out a bunch of water, right? The Krebs cycle is just like the Calvin cycle. It has a bunch of steps. This diagram doesn't even show all the steps other than the fact that we are taking our carbons and we're separating them to release carbon dioxide. In the process, we're getting a bunch of electrons. We're getting a little bit of ATP through substrate level phosphorylation. Again, that happens here and there, but it's not really the goal here. We really just want the electrons that we get in multiple steps. Ooh, and here's again, just to show you all the complicated changes. You can pause this if you want to look at that more. But then we have the glycolysis. Glycolysis is the first step, right? So I'm kind of going backwards because I was using kind of that idea. Hopefully that wasn't too confusing, or if it is, you can ask me questions. But glycolysis is needed to break down everything to get the pyruvic acid, what it says here, but pyruvate is the other name for it, right? Glycolysis is interesting because it's kind of its own process on its own. This is the very first way that organisms would have figured out how to get energy, right? It's very primitive. Going back to evolution, that every single organism uses glycolysis, which is interesting. And it happens in the cytoplasm, not inside of the mitochondria, like all the rest of the stuff is happening, right? This is a process where we take our glucose and we split it in half to make 2 3-carbon sugars. We do get some electrons that are going to go to the electron transport chain. We do get ATP. Although we did have to pay some ATP earlier, so we only get 2 total out of there. You don't really have to know about the energy investment and payoff steps too much anymore. So here's this diagram, ending kind of the way that we started before. We have glycolysis as the beginning out here in the cytoplasm. We're gonna strip some electrons from them. We're gonna get some ATP. But this is all just a processing step to get us to the Krebs cycle, also known as the citric acid cycle. This is also just a processing step where we strip electrons and make a little bit of ATP. The electrons are the stars into oxidative phosphorylation, where we just start pumping them through the electron transport chain to get a lot of ATP out of that, right? And so again, if you can explain this picture, just like the photosynthesis summary, then you're gonna be doing just fine. Some names for this one. The matrix is the inside part where the Krebs cycle happens, and then the intramembrane space is like the thylakoid version now. This is where the electron transport chain will be done, right? So maybe you'll have to know those names. A lot of times they come on diagrams though. There is one interesting thing about cellular respiration is that it requires the oxygen, right? And so if you don't have any oxygen, this causes a lot of problems because the oxygens can't accept electrons, which means that the NADH being made right here, which isn't working for me for some reason, there we go. The NADH made right there has nowhere to go. It's supposed to be dropping off its electrons on the electron transport chain to give to oxygen, but it can't do that. And if you have all these NADHs, you don't have NAD+, which means you can't do some of the reactions that happen right in here. And everything shuts down. So fermentation is a way to regenerate the NAD+ that you needed by taking our NADH and dumping the electrons into either alcohol or lactic acid, depending on if you are a human or a bacteria, right? We don't do alcoholic fermentation. We do lactic acid fermentation is what it's called, right? And so this is just, again, goes back to the early days of the ancient organisms that were gonna try to make make energy without oxygen, because oxygen didn't exist back then as free atmospheric oxygen. And so fermentation is the way to keep the energy flow, although you get way less energy out of it this way, right? And so there we have cellular respiration and photosynthesis. I know this was a long one and I'm talking really fast. I feel like I'm talking super fast. But if you have any questions, as always, hit me up on Hangouts or email me or some such method.